Welcome students. Today we discuss cell cycle checkpoints in our class. So now let's begin directly to the class and let's discuss the cell cycle checkpoints.
So we will start our class with a simple question. As cells move through the cell cycle, do they breach through the from one cell one phase to the next? That means is the movement of cell from one phase to the another phase of the cell cycle have any kind of any parameter checkpoints or anything else, or they can move freely? So if they are cancer cell, the answer is yes. They can move through the different phases of cell cycle easily without any kind of checking. But in case of normal cell, however, the cell moves through the cell cycle in a regulated way. They have to move through a regulated way. They use information about their own internal state and cues from the environment around them to decide whether to proceed with cell division or not. So they collect data from their environment, and after processing of the data, they decide whether they should move or not. This regulation makes sure that cells don't divide under unfavorable conditions, like if the DNA is damaged in a particular cell. When the DNA is damaged, that damaged DNA should not go into the next division phase, that should be restricted. In other case, when there is not room for more cells in the tissue or organ, and we call it a contact inhibition. When two cells are present in a close contact or close proximity contact, they are not able to divide. So what are the different cell cycle checkpoints? A checkpoint is a stage in the eukaryotic cell cycle at which the cell examines internal and external cues and decides whether or not to move forward with division. It's a particular point, it's a particular stage in the eukaryotic cell cycle where the cell have to decide based on its internal condition that whether they should move or not into the next phase of the cell division. There are a number of checkpoints, but most we will discuss the major three checkpoints, which is most important. The G1 checkpoint at G1S transition, G2 checkpoint at G2M transition, and the spindle checkpoint at the transition from metaphase to anaphase. So this is a pictorial view of the different checkpoints. So we will first discuss the G1 checkpoints, and in this particular checkpoint, cell actually look for the status of the nutrient, status of the growth factor, and any DNA is damaged or not. Next is the G2M checkpoint, and in this particular checkpoint, the cell actually look for the cell size and the DNA replication is completed or not. And the third one is the metaphase checkpoint, where the cell actually look for the chromosome spindle attachment thing. Chromosome spindle attachment. So in G1 checkpoint, this is the main decision point for a cell. That is the primary point at which it must choose whether or not to divide. Once the cell passes G1 checkpoint and enters into the S phase, it becomes irreversibly committed to the division. So upon completion of the G1 checkpoint, or upon successfully go through the G1 checkpoint, the next phase is the S phase, and this particular phase is irreversible. When a cell enters into the S phase, they cannot return from it. So that is why in this particular checkpoint, G1S checkpoint, cell have to decide whether they are moving or going forward or not. That is bearing unexpected problems such as DNA damage or replication error. A cell that passes the G1 checkpoint will continue the rest of the way through the cell cycle and produce two daughter cells. At G1 checkpoint, a cell check whether internal and external conditions are right for the division or not. So what are the different internal and external conditions? They are like this. Number one, size. Is the cell large enough to divide? So if the cell is not large enough or not mature enough to go for a further replication process, the cell will not move through the G1 checkpoint. Nutrients. When a particular cell is present in a nutrient rich condition, that means they are getting enough energy, or they have enough energy reserve or available nutrients for their division phase. So if they have enough nutrient, they will go through the G1S division, or else they will not pass through the G1S checkpoint. Third is the molecular signals. Is the cell receiving positive cues such as growth factor from neighbors? Yes, this is a very important thing, because when a particular cell receives the signals from its neighboring cells that yes, we are also in the same condition like you, then only the cell can move, because each and every cell of a particular location, a particular organ, a particular position of our body will grow in a similar pattern or a similar way. And fourth one is the DNA integrity, which is very important, because if the DNA integrity is not there, if DNA is damaged, the cell should not go for the replication process, or else that will create irreversible error, which may lead to several diseases. So these are not only factors that affect progression through the G1 checkpoint, and which factors are most important depend on the type of cell. So these are not the only factors. There are several other factors also, but all the factors depend on the cell type. For instance, some cell also need mechanical cues, such as being attached to the supportive network or extracellular matrix in order to divide. If a particular cell is not attached with the matrix, they will not divide. Like our body cell, muscle cells. In some cells, they are present in a suspension condition, like our blood cells. They are not supposed to require this attachment criteria. If a cell does not get the go-ahead cues it needs at the G1 checkpoint, it may leave the cell cycle and enter a resting state called G0. Some cells stay permanently in the G0 phase, while others resume dividing if conditions improve. That means this G0 state is a temporary condition. Now, what is the difference between G1 and G2 checkpoint? We should look into this before going forward to the G2 checkpoint. So the G1 checkpoint cell actually look for whether DNA is okay, the enough resource for cell is present or not, or that is required for the replication process. They build enough protein or not, or is the overall environment of the cell is okay or not. And in case of G2 checkpoint comes after DNA replication. So they will first check whether DNA is correctly replicated or not. If the DNA replication is complete or not, and thirdly, if the cell have enough protein or not. So in G2 checkpoint, to make sure that cell division goes so smoothly, the cell has additional checkpoint before M phase called G2 checkpoint. At this stage, the cell will check DNA integrity. Is any of the DNA damaged or not? And DNA replication, was the DNA completely replicated after the S phase or not? If error or damage are detected, the cell will pause at G2 checkpoint, and that will allow cell to repair itself. If the checkpoint mechanisms detect problem with the DNA, the cell is halted. The halted for that particular amount of time which is required to repair the cell. and the cell attempts to either complete DNA replication or repair the damaged DNA. So once they are able to complete the replication or able to repair the damaged DNA, they can further proceed through the M phase. But if the damage is irreversible, that means the cell is not able to repair itself. That means on that particular point of time, the cell will undergo apoptosis, which means the programmed cell death. Or this is a self-destruction mechanism, ensures the damaged DNA is not passed on to the daughter cell. And this is important to prevent cancer as well as several other diseases. So now, when we come to the M checkpoint, this M checkpoint is basically based on the spindle checkpoint. That means whether the spindles are properly created or not. This is also known as a spindle checkpoint, where cell examines whether the sister chromatids are correctly attached to the spindle microtubules or not. If sister chromatids are not correctly attached and say they undergo the process of completion of cell division, the cell may produce inequality in chromosome number, and that also leads to the detrimental diseases. Because the separation of the sister chromatids during anaphase is an irreversible step, the cycle will not proceed until all the chromosomes are firmly attached to at least two spindle fibers from opposite pole of the cell. So how does this checkpoint work? It seems that cells don't actually scan the metaphase plate to confirm that all the chromosomes are there. So cells actually do not have the ability to check the metaphase plate. Instead, they look for straggler chromosomes that are in a wrong place, which is much easier. It is better to find a wrong chromosome which is not supposed to present in a place when the other chromosomes are present in a metaphase plate condition. If the chromosomes are misplaced, the cell will pause mitosis, allowing time for the spindle to capture the stray chromosome. When the spindle is able to capture the stray chromosome, the mitosis can proceed. But if they are not able to capture the stray chromosome, again the cell undergo the process of apoptosis. So these are the three different checkpoints of cell division. Now we will come to our next part of the topic, that is the role of meiosis in life cycle of organism. Do you ever wish to that you could clone yourself so that you can do extra work, you can do extra study? So as because we are human, that is not possible for us to clone ourselves. But if we are a starfish or a cactus, we can do that. Some starfish are there who are able to clone themselves by just breaking an extra arm, which will then regrow into a complete starfish. That is just a picture like this. This is a complete starfish, and these are the starfish which are growing from an extra arm of another starfish. So they are genetically identical. So they are clone of themselves. And similarly, this cactus is also have the similar ability. This cactus, each and every lobe of the individual cactus are genetically identical of each other. So these reproductive strategies are example of asexual reproduction, which produces offspring genetically identical to the parent. In contrast, many plants, animals, and fungi produce offspring through sexual reproduction. We already have idea about the asexual reproduction and sexual reproduction. In our previous classes, we have already gone through this kind of thing. In sexual reproduction, sex cells from two parents combine in a process of fertilization, leading to the formation of a new genetically distinct individual. And some organisms, including the starfish and cacti in the example above, can actually reproduce in either a sexual or an asexual mode. All sexually reproducing species have certain key life cycle features in common, such as meiosis and fertilization. So fertilization is actually the process of fusion of haploid gamete to form a diploid cell called zygote. So type of sexual life cycle, there are three main type of sexual life cycle. That is, in a diploid dominant life cycle, haploid dominant life cycle, and in a diploid dominant life cycle, the multicellular diploid stage is the most obvious life stage, and the only haploid cell are the gamete. So that we call the diploid dominant life cycle. Like us, like human, our haploid cells which are present in our body or system is only the gametes, or otherwise we are a diploid organism. So diploid dominant means when the diploid stage is dominant over the haploid one. The second one is the haploid dominant life cycle. The multicellular or sometimes unicellular haploid stage is the most obvious stage and is often multicellular. In this type of life cycle, the single cell zygote is the only diploid cell. Fungi and some algae have this type of cell cycle. That means in this particular case, haploid cells are dominant throughout their life cycle, and diploid cells are only formed in the process of zygote formation. So diploid dominant life cycle, nearly all animals have a diploid dominant life cycle in which the only haploid cells are the gametes. Early in the development of the animal embryo, special diploid cells called germ cells are made in the gonads. That is the testes or ovaries of the matured individual. Germ cell can divide by mitosis to make more germ cells, but some of them undergo meiosis, making haploid gametes. Fertilization involves the fusion of two gametes, usually from different individuals, restoring the diploid state. So this is the overall life cycle. That is, this male and female are the diploid dominant organism, but they have a testes and ovary where they are able to produce haploid cells, which are the gamete cells. So when these two gamete cells are combined with each other, and these gamete cells are actually producing by the process of meiosis, and when these two gamete cells like sperm and ovum are fused with each other in the process of fertilization, they are able to produce zygote, which is the diploid cells. And once the diploid zygote are produced, they are undergoing the process of mitosis to produce a mature individual. So this is the diploid twin or diplodominant stage of the individual. So haploid dominant life cycle, most fungi and some protists, that is the unicellular eukaryotes, have the haploid dominant life cycle in which body of the organism, that is the mature or ecologically important form, is haploid. An example of a fungus with a haploid dominant life cycle is a black bread mold, whose sexual life cycle is known as a diagram below. In sexual reproduction of the mold, hyphae from two compatible individuals first grow towards each other. Where the hyphae meet, they form a structure called the zygosporangium. A zygosporangium contains multiple haploid nuclei from the two parent within a single cell. The haploid nuclei feast to form diploid nuclei, which are equivalent to zygote. The cell containing the nuclei is called zygospore. So these are kind of a primitive kind of approach, but these are mainly dominant over the fungi and algae. The zygospore may stay dormant for long periods of time, but under the right condition, the diploid nuclei undergo meiosis to make haploid nuclei that are released in single cells called spores. So this is the life cycle of the haplodominant organism. So this is the zygospore. The zygospore will undergo the process of meiosis to form spore. Zygospores are dominant, spores are haploid, and haploid spores are then undergo the mitosis to form hyphae. The two different hyphae may mate with each other, form a zygosporic structure, which contain multiple nuclei. The multiple nuclei are going to fuse with each other which we call a nuclear fusion, and form a zygosporic structure. This is the upper part is the haploid diplodominant part, and diploid part and lower part is the haploid part. And as we just go through this phase, we look at the after by looking at the picture, we are able to see that the haploid part is a part of the life cycle is much longer in this particular case than the diploid organism. So this is the end of our today's class, and in next class in the genetics we will come up with a new topics. Thank you.
If you like the video, please subscribe the channel and hit the bell icon. Thank you.